Welcome to another IB Environmental Systems and Societies video. Today we're going to explore the higher level content of topic 2.3 Biogeochemical cycles, diving deeper into how carbon, nitrogen, and other essential elements move through Earth's systems. Let's get into it. Let's start with the lithosphere, the solid, rocky outer layer of Earth that contains important carbon stores. The lithosphere includes both fossil fuels and carbonate rocks like limestone. These carbon stores took millions of years to form, and they play an important role in the global carbon cycle. Looking at fossil fuel formation, we see that it required very specific conditions in past geological eras. Most coal formed during the Carboniferous period between 359 to about 299 million years ago, when vast swamps and forests provided abundant plant material in warm, wet conditions. Oil and natural gas formed mainly in the Jurassic and Cretaceous periods between 201 and 66 million years ago, and those were formed from marine organisms buried in fine sediments under shallow seas. One of the major carbon stores in the lithosphere is limestone. It forms through a three-step process. First, calcium carbonate precipitates from seawater to form calcite, often in the form of marine organism shells. Then, these shells settle to the bottom and they become compacted on the ocean floor. Finally, with increased pressure and temperature over time as these layers are buried deeper and deeper, these deposits transform into limestone rock. One thing you need to understand when studying carbon stores is the idea of residence time. That's the average period a carbon atom remains in a particular store or reservoir. Without human interference through activities like mining, carbon in the lithosphere would have a residence time measured in hundreds of millions of years. But our extraction of fossil fuels has dramatically accelerated its release, rapidly returning millions of years of stored carbon to the atmosphere in just a few hundred years. Reef-building corals and mollusks play a special role in limestone formation through their calcium carbonate structures. As we just saw a few moments ago, when these organisms die, their hard parts can become fossilized and contribute to limestone deposits over time. Looking at a coral reef, we can see the living limestone builders in action. The calcium carbonate skeletons of corals accumulate over thousands or millions of years to create massive reef structures. These biological processes eventually form limestone deposits, and those limestone deposits are Earth's largest carbon reservoir. Limestone can also form through non-biological processes. Calcium carbonate can precipitate directly from water when the conditions change and are just right. This usually happens through evaporation, changes in water temperature or pressure, or shifts in pH level. Similar processes create cave formation like stalactites and stalagmites, and they also form travertine around geothermal springs like the ones you see here. In past geological eras, different types of organic matter underwent distinct fossilization processes. Plant material in swamps and forests became coal through one pathway, while marine organisms in ocean sediments became oil and natural gas through another pathway. The type of fossil fuel formed depended both on the starting material and the environmental conditions. Fossil fuel deposits formed during specific geological periods when conditions were just right. The Carboniferous period, named for its abundant coal deposits, provided perfect conditions for coal formation with its warm, swampy environments. Several million years later, the shallow seas of the Jurassic and Cretaceous periods created ideal conditions for oil and gas formation from marine organisms. Moving to another important part of the carbon cycle, we need to understand the role of methanogenic bacteria. Methanogenic bacteria are specialized microorganisms that produce methane as a byproduct of their metabolism, but only under anaerobic conditions. Those are environments lacking oxygen. These electron microscope images show the bacteria responsible for methane production. These methanogenic bacteria thrive in several types of oxygen-poor environments: swamps and wetlands, flooded rice paddies, and notably in the digestive systems of ruminant animals like cattle. Each of these environments provides the anaerobic conditions these bacteria require for methane production. Remember, anaerobic means without oxygen. Methane's behavior in the atmosphere is distinct from other greenhouse gases. It has a relatively short residence time of about 10 years before being oxidized to carbon dioxide. This is particularly important for understanding methane's role in climate change because methane is much more effective at trapping heat than carbon dioxide. The methane cycle involves both natural and anthropogenic or man-made sources. On the natural side, we see contributions from wetlands, from the ocean floor hydrates, and from forest fires. Human activities add methane through fossil fuel extraction, waste management, and agriculture, particularly rice cultivation and livestock farming. All this methane eventually oxidizes in the atmosphere through various chemical pathways to become CO2. Check out these global methane measurements over time. You can see a really clear upward trend. These measurements, which were all taken at monitoring stations around the world, show how atmospheric methane concentrations have increased significantly since the 1980s. Notice how the rate of increase is sharper in recent years. That means we're adding more carbon to the atmosphere faster. The nitrogen cycle is another important biogeochemical process, and it also has distinct organic and inorganic stores. Organic nitrogen stores include proteins and other nitrogen-containing compounds in living organisms and dead organic matter. Inorganic stores include atmospheric nitrogen gas and various nitrogen compounds in soil and water. This diagram shows how nitrogen moves through different stores in an ecosystem. Notice how the cycle connects atmospheric nitrogen to soil nutrients and then to plants and animals, and then back again. Each transformation requires a specific set of conditions and often involves specialized bacteria. Speaking of bacteria, they play essential roles throughout the entire nitrogen cycle. Different types of bacteria are responsible for key transformations that make nitrogen available to living organisms. Without them, there is no nitrogen cycle on Earth. Let's trace these bacterial processes. Nitrogen-fixing bacteria transform atmospheric nitrogen gas into ammonia, which makes the nitrogen available to plants. Nitrifying bacteria then transform ammonia into nitrites and ultimately into nitrates. Then we have decomposer bacteria break down organic nitrogen compounds back into ammonium through a process called ammonification. The cycle continues with denitrifying bacteria, which convert nitrates back into atmospheric nitrogen gas under anaerobic conditions. This completes the nitrogen cycle and maintains the balance of nitrogen in Earth's different systems. However, human activities, particularly agriculture and industry, have pretty significantly altered these natural cycles through the use of synthetic fertilizers and other interventions. Let's look at denitrification in more detail. Denitrification only occurs in anaerobic conditions, particularly in waterlogged soils where oxygen is absent. In these anoxic environments, specialized bacteria convert nitrates back into nitrogen gas through a series of steps, first reducing nitrates, NO3, to nitrites, NO2, then to nitrogen monoxide, NO, and nitrous oxide, N2O, and then finally back to diatomic nitrogen gas, which is N2. This process completes the nitrogen cycle, returning nitrogen gas from the lithosphere in soils back into the atmosphere. An interesting adaptation to these low nitrogen conditions can be seen in certain plants like pitcher plants and sundews, and those plants have evolved to capture insects as an alternative nitrogen source. Plants face a significant challenge. They can't directly use atmospheric nitrogen. However, nature has developed an elegant solution through mutualistic relationships between plants and nitrogen-fixing bacteria. These beneficial partnerships allow both organisms to thrive. The bacteria provide fixed nitrogen that plants can use, while the plants supply carbohydrates to the bacteria. Here's a classic example of this mutualism: legumes and their rhizobium bacterial partners. These edamame or soybean pods represent the legume family, which forms specialized root nodules that house the rhizobium bacteria. This partnership is so effective that farmers often use legumes in crop rotation to naturally replenish soil nitrogen between plantings of more demanding crops like corn or wheat. Another remarkable example is found in acacia trees, which form relationships with multiple nitrogen-fixing bacteria species. These acacia trees dominate nutrient-poor savannas precisely because of these partnerships. Not only do they enrich the soil with nitrogen, but they also provide vital nutrients to browsing animals like giraffes while offering shade in hot environments. Alder trees demonstrate yet another variation of this mutualistic relationship, partnering with Frankia bacteria species. As pioneer species, alders are often among the first trees to colonize disturbed sites. Their nitrogen-fixing abilities help facilitate ecological succession by enriching poor soils, making it possible for other species that need more nutrient-rich soils to eventually establish themselves. The nitrogen cycle involves both transfers and transformations, which you can see in this diagram. If we start with mineral uptake by producers, we can follow the movement of nitrogen through various biological processes. Plants absorb nitrogen compounds through their roots. They incorporate them into organic molecules through photosynthesis, and then these compounds move through food webs via consumption. When organisms die or excrete waste, decomposition and ammonification return that nitrogen to inorganic forms. This systems diagram helps us visualize these complex flows. Notice how nitrogen moves between different stores, from atmospheric nitrogen gas through various transformations in soil, then to nitrogen compounds in producers and consumers, and then back again to the atmosphere. The red arrows indicate transfers, while the blue arrows show transformations. Each process, whether carried out by bacteria or plants, plays a really important role in maintaining the nitrogen cycle. Human activities have dramatically altered natural nitrogen cycling. We're going to examine 4 major impacts: deforestation, agriculture, aquaculture, and urbanization. Each one disrupts different aspects of the nitrogen cycle in different ways. When forests are cleared, several disruptions occur simultaneously. The removal of trees reduces nitrogen uptake from soils, and that leads to increased nitrogen loss through erosion. Additionally, the disturbance of soil structure disrupts those microbial communities that are responsible for nitrogen cycling. Agricultural practices might have the most profound impact on nitrogen cycle of any of these. Intensive farming, especially monocultures like this one, often relies heavily on synthetic fertilizers, while soil compaction from heavy machinery can affect bacterial activity. Irrigation changes soil conditions, and monoculture farming reduces natural nitrogen fixation that would typically occur in more diverse plant communities. Aquaculture introduces its own set of challenges to nitrogen cycling. Fish farms concentrate waste production in small areas, leading to nutrient enrichment of surrounding waters. The addition of feed and various additives further alters natural nitrogen cycles in aquatic systems. In urban environments, the natural nitrogen cycle faces multiple disruptions. Impermeable surfaces increase nitrogen runoff, while concentrated human waste creates localized nitrogen hotspots. Look at this picture. It's all concrete where nothing penetrates. And think of the number of people living in these buildings and the number of toilets they're flushing every single day. That's a lot of nitrogen concentrated in a very small space. Vehicle emissions also add nitrogen oxides to the atmosphere, and the sealing of soils prevents natural cycling processes. Combined, these human activities lead to significant changes in the nitrogen cycle. Excess nitrogen disrupts aquatic ecosystems and contributes to air pollution, especially urban smog. Our actions are shifting the complex balance of flows within the nitrogen cycle, disrupting equilibria that have been established over millions of years of evolution. The Haber process represents one of humanity's most significant interventions in the nitrogen cycle. This industrial process converts atmospheric nitrogen into ammonia for fertilizer production, fundamentally changing how nitrogen moves through Earth's systems. You don't have to know all the different steps of the Haber process, but you should understand the essential elements of it. The Haber process takes nitrogen from the atmosphere, where nitrogen makes up 78% of our air, and combines it with hydrogen under specific conditions of temperature and pressure. The result is ammonia, which becomes a key ingredient in commercial fertilizers. While this process has revolutionized agriculture and helped feed billions of people, it comes with pretty significant environmental costs. The Haber process presents us with a complex trade-off. On the positive side, it enables large-scale fertilizer production, and that dramatically increases crop yields and helps feed our growing global human population. However, these benefits come at a pretty high cost. High energy consumption, significant CO2 emissions, contribution to water pollution, and soil acidification. Perhaps most concerning is how it creates dependence on industrial fertilizers while potentially damaging natural nitrogen cycling processes. The accumulated effects of human nitrogen use have now pushed us beyond the planetary boundary for the nitrogen cycle. This means we've exceeded Earth's capacity to maintain a stable nitrogen balance or equilibrium, potentially leading to irreversible changes in Earth's ecosystems. Look at the Stockholm Resilience Center's planetary boundaries diagram, which we first saw back in ESS topic 1.3 sustainability frameworks. You can see that biogeochemical flows, particularly nitrogen, have moved well into the high-risk red zone. Several key indicators demonstrate this crossing of the boundary. Atmospheric nitrous oxide levels are now significantly higher than in pre-industrial levels. We're seeing widespread coastal dead zones that are caused by excess nutrients, we've got declining biodiversity and nitrogen-enriched ecosystems, and there's been an alarming increase in harmful algal blooms. Rising nitrate levels in groundwater and changes to soil microbial communities provide further evidence of this kind of disruption. Given these serious challenges, global collaboration has become essential to address the use of nitrogen in industry and agriculture. We need coordinated efforts to bring the nitrogen cycle back within planetary boundaries. Agricultural reform represents a key area for improvement. By implementing precision agricultural techniques, we can optimize fertilizer application and reduce excess nitrogen use. Promoting organic fertilizers and crop rotation helps restore natural nitrogen cycling. Planting or maintaining buffer zones along waterways can also prevent nitrogen runoff, while additional sustainable farming practices support long-term soil health. Policy changes form another important part of nitrogen management. These include setting stricter limits on nitrogen emissions, implementing nitrogen pollution taxes, and requiring improved waste management practices. We can look to successful examples like the EU Nitrates Directive, China's zero growth in fertilizer use policy, and Denmark's nitrogen quota system as models for effective regulation. Industrial changes must also play a role in addressing disruptions to the nitrogen cycle. This includes improving the efficiency of the Haber process, developing alternative fertilizer production methods, and enhancing wastewater treatment. Promising innovations include green ammonia production that uses renewable energy, plasma-activated water for fertilizer production, and biological nitrogen removal systems. These changes illustrate how we can work to bring the nitrogen cycle back into balance through coordinated action at multiple levels, from individual farms to international agreements. Success will require continued technological innovation, policy reform, and global cooperation. That's it for the higher level content about biogeochemical cycles. Remember that the biogeochemical cycles that sustain life on Earth are remarkably resilient, but they do require careful stewardship. Until next time, happy learning.